Hello everybody, today I will explain you about class 9 chapter 5, that is the fundamental unit of life. Yes friends, we all know that the fundamental unit of life, that is basically a cell. Cell, it is small component which is present in every citizen's body, and it is known as the structural and the functional unit of life. Why so? Why so it is known as the structural and functional unit of life? As it is known as structural unit of life because cells forms tissue forms organs, organs form organ system, and organ system forms finally a body. That's why cell is known as the structural unit of life. But why so functional? Because in unicellular organisms there is a single cell only present, and throughout the life they have only a single cell. So this single cell performs every activity in a cell, in a body. Like in amoeba, it is a unicellular cell, but only a single cell can control whole body. As we are multicellular, our millions of cell controls our body, but in this amoeba a single cell controls our body. That's why it is known as functional unit of life. Largest cell is known as ostrich egg cell. Ostrich egg, this is ostrich egg, largest cell. And smallest cell, PPLO, that is pleuropneumonia like organisms, and it is mycoplasma. The size and shape of the cell depends, the function depends on the size and the shape of the cell. Like RBCs are discoidal shapes, they do not have a nucleus. Like nerve cells are long cells. Nerve cells, nerve cells are long cells because they have to transfer messages. That's why they are long. So the function depends upon the shape and size of the cell. Please provide me that book. Please provide me that book. Please provide me that book. Yes, friends. Then comes who discovered the cell. Firstly, the cell was discovered by Robert Hooke in year 1665. He observed a dead cell, that is Koch cell, in his own microscope. Later on in 1674, Anton van Leeuwenhoek discovered living cell. Then protoplasm was discovered by Dujardin, but the name given to protoplasm is by Purkinje. In the year 1838, a scientist named Sliden, and 1839 scientist named Sean, they both gave cell theory. Now the cell theory stated that all plants and animals are made up of cells. Cell is the basic unit of life. Cells arise from their pre-existing cells. Later on, in 1885, Rudolf Virchow stated omni cellula a cellulae. This means that cells arise from their pre-existing cells. In a cell, there is division of labor is seen as in a particular cell there is mitochondria present, Golgi apparatus is present, nucleus is present, so they all have their particular functions to do. That's why in cell there is division of labor seen, like in our body. In our body also division of labors is seen as like hearts pumps blood. That's so. Then comes that what is cell made up of? Cell is made up of three components, namely nucleus, then cytoplasm, plasma membrane. Let's discuss about plasma membrane. What is plasma membrane? It is the outermost covering in case of animal cell. But it is not outermost covering in case of plant cell, because they have an addition to it, that is cell wall. So that's why it is the second most covering in the plant cell, but it is outermost covering in animal cell. It allows only specific materials to go in and out of the cell, like H2O molecules inside, CO2 molecules outside, like somewhat like this only. Then here a question comes that how does the movement of substance takes place into a cell? How do substance move out of the cell? See, imagine that this is a cell. Here inside CO2, oxygen, CO2, oxygen must be present. Outside also CO2 is oxygen, CO2. oxygen are present like so. Here inside CO2 is an excretory substance that should be moved out of the cell. So in here CO2 has a higher concentration, and here CO2 has lower concentration. So diffusion of term, that means that movement of substance from the higher concentration to the lower concentration. So in the cell CO2 has higher concentration, and outside the cell CO2 has lower concentration. So in this way CO2 will move outside the cell. In O2 have lower concentration inside and higher concentration outside. That's why O2 will move inside the cell. The next topic comes that what is hypertonic solutions. Hypertonic solutions, isotonic solutions. In hypertonic solution, here a cell is placed. Hypertonic and hypertonic solutions, the water outside the cell has higher concentration at that of inside of cytoplasm. Means outside water of this cell is low as compared to the cell. So that's why the water inside the cell will move outside. That's why cell will shrink. In case of plant cell wall will not shrink, only inside part will shrink. This is known as plasmolysis. In hypotonic solutions, the concentration of water outside the cell is low. Means there is more water outside the cell than compared to inside of cytoplasm. That's why cell will move inside. In case of plant cell, it will be known as plasmolysis, and in case of animal cell, it will burst. Isotonic solution, no change will be occurred in this because the concentration will be equal to that of cytoplasm. So let's revise. Hypertonic solution means outer concentration is higher, means water is low as compared to cell. Hypotonic, outer concentration is lower, means water is higher. Then comes what is cell wall. It is only present in plant cell. Why so? Because plants are stable. So to protect them, cell wall is present. Then imagine that this is a cell. Cell wall of two substances are joined by middle lamella. Calcium plus magnesium pectate. This was all. The cell wall maintains the shape of the cell as it is permeable. As it is permeable, it allows entry of any substance. Cell wall maintains the shape of the cell. And comes the main part of this chapter, that is what is nucleus. nucleus. It is known as the headquarter of the cell. It controls all the function. It also helps in transfer of hereditary characters from the parent to the offspring. In a cell, there is a nucleus in which chromatin material is there and DNA. Chromatin material is an entangled mass of DNA plus proteins. Where during the time of cell division, these chromatins are like this, forming chromosomes. They become straight during the time of cell division, but they are like this during none of the activities. Then nucleus, here comes that in nucleus that genes are present, and due to genes only it is the transfer could happen. The basic unit of DNA, that is known as genes. The basic unit of DNA is known as genes. Now next comes cytoplasm. Cytoplasm is a fluid material inside the cell which floats all the cell organelles and nucleus too. And cells are enclosed membranes. In prokaryotic cell, beside absence of a defined nuclear region, membrane-bound cell organelles are all also absent. On the other hand, eukaryotic cells have nuclear membrane as well as membrane enclosed organelles. So, in a bacterial cell, the nucleus is not well defined, so it is known as nucleoid. In our cells, that is eukaryotic cells, nucleus is defined, so nucleus only. There is a process known as endocytosis, through which amoeba engulfs foods. Then let's discuss about various cell organelles. First comes the endoplasmic reticulum. Endoplasmic reticulum is a large network of membrane-bound tubules, sheets. This is a long type tubules or oblong bags, or known as vesicle bags. There are of two types, SER and RER. Smooth endoplasmic reticulum, rough endoplasmic reticulum. As name only defines us that its surface, RER surface, is rough due to the presence of ribosomes. Here ribosomes are not present, and ribosomes are known as protein factory. That's why RER synthesizes proteins and SER synthesizes lipids that is present in adipose tissue, fats molecules. Let's rewind it. Plasma membrane is made up of proteins plus lipids, and SER and RER. SER gives lipids and RER gives proteins. That's why it also helps in the formation of plasma membrane. The function of ER is to serve as a channel for the transport of materials, especially proteins, between various regions of cytoplasm or between cytoplasm and the nucleus. ER also functions as a cytoplasmic framework, providing some for biochemical activities of the cell. SER plays an important role in detoxifying drugs, poisonous things. Comes next that cell organelles, that is Golgi apparatus or Golgi body. Here the cisterns are present parallel to each other. Here packaging is done, synthesizing is done, and manufacturing is done in Golgi apparatus or Golgi body. It is located near endoplasmic reticulum. Like as so, from endoplasmic reticulum, proteins will be sent. So here, packaging of proteins will be done, and due to packaging of proteins, it will be giving proteins to all the cell, means where it is needed. The protein where it is needed, it will also provide protein and helps in the formation of plasma membrane. The main function of Golgi body, because through Golgi body only lysosomes are formed. Here a question comes that what are lysosomes? What are lysosomes? Then lysosomes are a kind of waste disposal system. Means like lysosomes helps in the clean of drugs. It also digests foreign materials, drugs like type things. Then lysosomes are known as suicidal bags. Suicidal bags, because in lysosomes there are 40 hydrolytic enzymes are present. Hydrolytic enzymes. In during means when cell is sometimes then disturbance in a cell occurs. So that's a lysosomes burst. So their hydrolytic enzymes move out of the lysosomes and digest our own cells. That's why due to this they are known as suicidal bags. Comes another part, that is mitochondria. Mitochondria, it is something like this. Here oxysomes are present. These are known as cisterns, or the turn like this is known as a cisterns, and in cisterns the oxysomes are present. These are known as oxysomes or F0 F1 particles. Due to this only they synthesizes ATP, adenosine phosphate, adenosine triphosphate, sorry. Okay, then or energy. In mitochondria, DNA is also present. Like mitochondria has their own DNA. There are two autonomous organelles in a cell, that is mitochondria and plastids. There are two autonomous organelles, semi-autonomous or autonomous organelles, that because they synthesizes their own, because they have their own DNA. Then comes what are plastids. Plastids firstly only present in plant cell. Only present in plant cell. There are three types: chloroplast,